Let's go. Hello, my dear friends. Best greetings from Kyiv, the capital of Ukraine. My name is Fursov. I am a research entomologist, beekeeper, and teacher. I am working as a senior research scientist at the Institute of Zoology of National Academy of Sciences of Ukraine in Kyiv, the capital of Ukraine. I am studying small parasitic wasps. Parasitic wasps, which are separated from all other Hymenoptera groups. Hymenoptera parasitica, because Hymenoptera, this is a very large group of insects, which includes ants, bees, bumblebees, sawflies, and parasitic and phytophagous wasps of different families and different superfamilies. We can use this book just for identification of Hymenoptera groups, for differentiation of different biology features, and we will be talking today about parasitic Hymenoptera, or group of Hymenoptera parasitica, because this is the most interesting, from my point of view, group, and which is interesting for me as a specialist, because I'm studying the taxonomy, biology, and I'm studying the group which is called family Trichogrammatidae, and which is indicated here on this poster. And here there are several other groups of CHALCID wasps of a superfamily Chalcidoideae, including Encertidae, Pteromalidae, Eurytamidae, Tetracampidae, Maimaridae, Trichogrammatidae, Eulophidae, Aphilinidae, and others. They're all here just in the end. They're very different. There are huge variety in their biodiversity, and they're spread all around the world except Antarctic. They're living even in Arctic, but not in Antarctic, too cold. But there are a lot of species distributed in desert areas, in semi-desert areas, in a tropical region, in a semi-tropical region. So the biodiversity in tropical and semi-tropical regions studied not so good, because it's quite difficult to access some areas and biodiversity of plants, and variation of environmental habitats changing drastically, reducing during the last century. So the number of species is decreasing, but nevertheless there are some remnants of natural habitats in these areas. In every country it's the same situation, quite difficult one, but nevertheless in native areas it's possible to find Hymenoptera parasitica and describe them. And this is the obligation of parasitologists and hymenopterists and scientists who are studying Hymenoptera parasitica, to describe new species of Hymenoptera parasitica, to discover their biology. Describe biology, and describe their morphology as new species, or describe the distribution of already known species. And for that, a professional entomologist using different scientific journals, peer-reviewed journals as now it's called. They're publishing books, they're publishing papers, scientific papers in such journals like this: American Entomological Society, Transactions of Entomological Society, like in the book, Journal of Natural History in Britain, and Proceedings of Royal Entomological Society in Britain. In Ukraine, we are publishing the papers, scientific papers, in Journal of Ukrainian Entomological Society, and definitely accomplishing hard work of identification and collection and sorting, identification of all different species. Entomologists can publish different books describing their biology and correlations between hosts and correlation between parasitoids. Sometimes we are publishing even atlases, like this atlas published in our department in Kyiv, Atlas of Entomophagus insects, about Hymenoptera parasitica, including superfamily Ichneumonidae and Chalcidoideae, and some other predaceous insects as entomophagus insects. So here, atlas with drawings of different pictures, different families and different species. But now we will also can make it different photographs of insects for publishing in scientific papers, for identification and for description of morphology, which is used for correct identification. We can use also today for showing you just some features of morphology. I can show you some videos and some photographs. I can use this book of Insects and Arachnids of Canada to read you a little bit more details about their morphology. And let's start from the beginning to show you some views of different families, different species of Hymenoptera parasitica under the microscope, under the high magnification. But for beginning, I will show you the most pleasant insects in the Hymenoptera group, which is where you are familiar about it. So bumblebees. All people enjoy bumblebees. If you show bumblebee, people are laughing and enjoying the situation. And this is a worker of a bumblebee. You can see because this is a worker, it is not a drone. On the hind, like we see scopula, very shining hind legs, which is adapted just for collection of pollen. And if you can find bee, which is just deceased or weak, or just stopped after the rain, somewhere on flower, you can bring it to home and feed it just even on your palm or just on your finger or just on a plate, given just a drop of honey or drop of sugar liquid, and honey bee and bumblebee will be very happy about it. But bumblebee, little bit more relaxed, they're not so worried about situation where will not escape immediately as a honeybee, but bumblebee will stop and feed on honey, so you can train bumblebee to be friend and feed bumblebee with drops of honey. But after feeding, it's better to release bumblebee, because bumblebee is very useful, beneficial pollinator, coming to different flowers, like here. Sleeping drones probably came just inside the flower of malva. Flower is quite big, so with some pollen, and so you see very tiny drops, or quite rather big drops on the body of bumblebees, and even some drone, some other small Hymenoptera came. So you can see how they're pollinating different flowers. On the body, they are full of different pollen. And let's start to watch parasitoids under the microscope. Here we can watch CHALCID wasps, group belonging to the family Encertidae. Encertidae family, very large group of insects in the superfamily Chalcididae. So we can see here just some insects under the small magnification of microscope, and then under the biggest magnification of microscope. So you can see here quite a robust shape of body, with some a little bit elongated antennae for males and shorter and hairy antennae, and shorter antennae for females. This is a female. Very special structure of thorax, very special unique structure of abdomen. The body generally compact, but some species are very flattened to strongly even flattened forms, and some species even ant-like forms. Mesopleuron on the both sides of the body, shining with a very fine sculpture, very enlarged and inflated, without special impressed lines or grooves. And mesopisternum and mesopimeron not differentiated for Encertidae family, which is differentiated them from the family Eupelmidae. Prepectus divided into two triangular scleroids. Midcoxae usually widely separated from hind pairs and inserted slightly or in front of a midline of a mesopleuron. So the midcoxy inserted very far strongly to the front part of a body. Just let's watch it under the high magnification of microscope. Some species are pteros, some brachypteros, but many species are fully winged. But marginal vein is very short instead of a very short submarginal vein. Here we see enlarged mesopisternum, so like a plate on both sides of the body. Here just darkened wings, curved shape of hind legs, and very compact antenna with some inserted mandibular pulpa. And very special citation on abdomen. Species have a very wide trophical relations with different hosts, including parasitizing Homoptera, Coccoidea, and parasitizing different groups, including some larvae of mites, or to be precise, Ixodidae ticks. But trophical relations we can discuss later in separate video, because trophical relations of Encertidae is really very wide. Let's watch other families of CHALCID wasps. And here, yes, okay, this one. We watched this group of Encertidae, and here several groups of CHALCID wasps under the microscope, black colored and with very elongated wings, elongated antennae. These are tiny parasitoids of family Mimaridae. On the right side, the down part of, and the left part, so separated. The very smallest of another genus are Laptus or Anagrus, which are really very small parasitoids of eggs of leafhoppers, and most of species of the family Mimaridae were egg parasitoids. Some eggs of insects were of bigger size, some are very small, like here, very tiny in the central part. Parasitoids were smallest, even, you see, several times smaller than the biggest. The smallest here, about half of millimeter, 0.5 millimeter size of Mimaridae family. They can be easily collected in yellow pen traps, in malaise traps. But the best material which is used for identification and for study trophical relations is to rear them from host species of other plant, of other insects. For that it needs to collect eggs of insects, keep them in plastic or glass tubes, better in glass tubes, and study them daily under the microscope to check the emergence of parasitoids, and you can find emerged parasitoids. Here we can see also some Encertidae on the top of a screen. This is a male of Encertidae family. Left part also Encertidae family, and right part a brown color also Encertidae family. I cannot say species because here a huge mixture from distant area, at least more than 5,000 species in Encertidae family and more than 2,000 species in Mimaridae family. So it's quite difficult for identification. For identification we need to make very tiny microscopic slides. You see here again Mimaridae on the right side, and in the center this is bigger one, Encertidae, and here's some species also belonging to tiny Aphilinidae probably. Aphilinidae, yes, of brown and yellow color here, some individuals in the center belonging to Aphilinidae family, which are quite often can be collected by yellow pen traps as very smallest insects, but they're easily collected and then sorted in yellow pen traps. So here again on the left part, Anagrus species, and the black one is Polynema species, and several genera of Mimaridae family. And on the left part of a screen, probably one individual of Trichogrammatidis family of a genus Oligoceta, left part of screen. So here, huge biodiversity of different species. Let's check Mimaridae under another microscope picture. It's quite possible to change magnification. Here we can watch them under the big magnification. Here very elongated wings, very long antennae. On the right part of the screen, even longest antennae. So this is a male with shortened abdomen. And the down part of screen, family Encertidae. This is Encertidae family, black colored, and brown colored. This is all 4 specimens of Mimaridae. So here, this is a selected, sorted part of collection, because in yellow pen traps and Malay traps we can collect many individuals of different families of parasitic wasps, and not only parasitic wasps. But after that you need to sort them to families, groups, to separate Eppendorf for plastic tubes or glass tubes for next preparation on microslides and for next identification. So here in the dark, in the center, this is an Encertidae family, and the down part, here some Aphilinidae, yes, here, with red eyes, 2 individuals with red eyes, and on the right part, 3 specimens of Aphilinidae with scutellum, with yellow scutellum, yellow part of a thorax, blackish, but with yellow part of thorax, and on the right part, very tiny, nearly whitish or nearly white body of a Mimaridae family. Again, one big one on the down part of screen, yellow Mimaridae, and brown to brown here, one Encertidae and one is Aphilinidae. Aphilinidae, many species have very tiny size, here about 1 millimeter and less. 1 millimeter and less, so they are difficult for identification. As a dry individual, all of them, they need very tiny preparation on microscopic slide and careful study under the optical microscope. Here on the left part of the screen, genus Mimar with very elongated wings, which are hairy with very long citation and black disc of wing. So this is genus Mimar. And one another individual, also Mimaridae family. Again, yellow one and black one of Mimaridae family. All parasitoids of eggs of different insects. And here, in the central part of the screen, this is a male of Mimaridae family with very long antennae. Here, central part, 3 individuals of a Encertidae family. Again, Mimaridae black one and Encertidae yellow one. So you see also quite a variation of different species, males and females of Mimaridae and Encertidae and Aphilinidae. and down part probably Aphelinidae. Black one and yellow one is Mimaridae. Okay, let's watch. This is Encertidae. Encertidae, very short marginal vein, very compact antenna, and tiny small yellow, the right side of screen, very tiny yellow colored Mimaridae. Okay, let's watch one another sample. Here, more individuals of other families. Here, this individual belonging to the family, this one, Pteromalidae, male, and the previous one was female, Pteromalidae family. Many parasitoids of caterpillars, parasitoids of pupae, of different moths and butterflies. Here, you need to watch antenna. Yes, also Pteromalidae family. Yes, female with very elongated and sharply top of abdomen. Here, Pteromalidae. So this is a sample with Pteromalidae family with flattened thorax, elongated. And here the species of Encertidae family with extended ovipositor. Here Pteromalidae family, again Pteromalidae family, you see with very well extended, developed mandibles on head. This is a male of Pteromalidae. Others. Here Pteromalidae family. Here, this one is Encertidae with, you see, Encertidae. Again, different species. Encertidae. Here a group of Encertidae. Male and female. Here with elongated body and elongated abdomen. You see, and very special structure of antennae. Elongated, very slender shape of body, and you see extended stick on the top of the abdomen. This is ovipositor. Ovipositor is extended. So probably one species, two, three species different, for sure, with different shape of antenna and different shape of ovipositor, and well segmented antenna, and especially enlarged and flattened mesopisternum sides of a thorax. So this, and this is Eulophidae. This is tiny Eulophidae, shining greenish color. So some Eulophidae. And here Eulophidae with, you see, with branches, antennae with branches. You see, not one segmented, but several branches of on antenna. And here Pteromalidae. Pteromalidae, and here again Pteromalidae with developed mandibles. Well, and the last here with extended ovipositor after death, Pteromalidae. And the last but not the least, I show the large magnification of insect. And here, next family which we have not seen today under the microscope in alcohol. This is photo of family Euphelmidae with swollen clava of antenna, rounded shape of a head, very elongated mesopisternum and metapisternum, or just abdomen. Abdomen is elongated. Here some light, elongated abdomen, and when many individuals of the family Euphelmidae die, they're just changing the shape of abdomen, and abdomen is turned back on the thorax. So it looks like U-shaped thorax, like here. For some individuals, abdomen just curved a little bit, but in many cases curved so much that dry insects becoming with very unusual shape of body, and also with very enlarged and flattened mesopisternum, which is separated on two different parts, with a groove in the middle part, and very flattened mesosoma, with very flattened mesosoma or thorax. So where some of these species, and probably this species, is parasitoid of wood-boring insects, the larvae of wood-boring insects. Some species are egg parasitoids. For Eupilmidae family, there is huge variety of hosts which these parasitoids are parasitizing. So not only wood-boring, but many different species, different groups of insects are parasitized by Eupilmidae family. So these are some groups of parasitoids, some groups of CHALCID wasps under the microscope. Not too many, not too much, but nevertheless some groups of very different shape of body. We learned today about Eupilmidae, Pteromalidae, Aphilinidae, Encertidae, Trichogrammatidae, Maimaridae. You see, huge biodiversity of parasitic wasps, which can be found in sample of insects collected using Malaise trap, using yellow pen traps, and for many of them, unfortunately, but the trophical relations, their hosts, their meal, where they're living, still unknown. For some species it's studied and it's known, but with many species of parasitoids, hosts unknown. So, and the study of biology of these insects can be very useful, especially study of eggs of different insects, because eggs are hidden in many cases. They are concealed in plants, in stems, in leaves, under the epidermis of plants. So they are a little bit invisible. If you can collect some plants with stems, with flower pods, with eggs of different insects, identify eggs, because from unparasitized eggs, just the larva of host will hatch usually. So it's quite visible. It would be just either cricket, grasshopper, or it can be bug, Homoptera group, or it can be Lepidoptera, larva, caterpillar, or it can be larva of beetle. So preliminary, every species of phytophagous insect is connected with more or less one or several species of their plant. So some phytophagous insects monophagous, some oligophagous. So, but nevertheless, on the same group of plants, you can collect hosts. So you can propose that this host probably is the host of this parasitoid. And if you can collect eggs of this host, keep them in glass jars or plastic tubes, in small on petri dishes, under the very careful study and checking under the microscope, you can rear, not by case, but by after very careful observations of these insects, you can rear from host eggs parasitoids. And these parasitoids can belong to different families. They can be Pteromalidae, Aphilinidae, Maimaridae, Eupilmidae, they can be Trichogrammatidae. And many species are still unknown, unstudied, and undescribed. That means they have never been mentioned in scientific literature. So if you are interested, you can be very careful about observation in the nature, and if you can find the eggs of insects, it can be Homoptera, Diptera, dragonflies, beetles, Lepidoptera, any kind of eggs of insects, you can keep them in plastic tubes, in glass tubes, separate them in small tubes, closed tubes, not with plastic cork, plastic cap, but with cotton for breathing, so your sample will be alive, because you can put here some tiny leaf with piece of laid eggs of Lepidoptera, with eggs of Homoptera, with eggs of beetles, and you can rear parasitoids from eggs of different insects. And this study, this observation can be very useful for biological purpose. So this biological feature of this or that parasitoid can be unstudied, but if you can bring this material or you can send this material for specialists, for instance, to our department in our institute, it can be different groups of parasitoids, but we have several taxonomists who can study these insects and who can identify these parasitoids and tell you precisely the name, who is this parasitoid, who was reared from eggs of Lepidoptera, beetles, butterflies, or other insects. And the precisely identified name can be used for scientific publication, for description, either in publication in one or another type of scientific journals. And it can be very useful for your career, for your scientific research, or for your study in your university. So let's study together different groups of insects. Let's study egg parasitoids, larval parasitoids, pupal parasitoids. But egg parasitoids are the most unstudied groups because it's difficult to collect them, and majority of received material will be really unique from biological point of view, unique from unstudied biology. So this is today's lecture about parasitoids, about their biodiversity. I hope you have seen some interesting insects, and if you are interested in some cooperation in the study of parasitoids, welcome to discuss. You can find my email under the description of this video, and I can show you my email directly so you can write it. So you can contact me on email for your private questions online. Ask your questions, write your comments, and if you have interest to be patron of my page, patron of my channel, you can find also Patreon page link and in the description of this video. So welcome to Patreon, to be sponsored as well, to donate for a cup of coffee. It will be pleasant feedback after watching of some funny, interesting videos which I collected for many years in different laboratories. To be continued, by the way, don't worry, this is not the last, but not the least. To be continued. See you next time on my stream. Write your questions, write your comments, and contact if you have private, interesting questions or proposals for collaboration communication. Thank you for watching. Good luck. See you soon. Someone who was Mr. Turchinsky wrote, good kids, good babies. Yes, they are very tiny because many parasitoids, we have a very tiny size of from 0.5 millimeters till maybe 2 millimeters. And 7 millimeters parasitoids are really very huge, considered to be very huge. Even in some groups, 1 millimeter in some families, this is a big one. And don't forget, Ukraine is forever. Good luck and see you soon on my channel. Ukraine is forever. Bye bye. See you soon on my channel. To be continued. Good luck.